Welcome to ICU Primary Prepcast. Hi, my name is Swapnil, and today we are going to cover another primary snippet around smooth muscle excitation contraction coupling. Now, with regards to smooth muscle, it is usually a non-striated muscle tissue and it causes movement under involuntary control, unlike to skeletal muscle. There are key differences with regards to structure, how the smooth muscle differs from skeletal muscle, especially with regards to excitation contraction coupling. And these differences include, first of all, T tubules, myofibrils, and sarcomere are absent in smooth muscle cells. Similarly, troponin, which is a regulating protein for contraction, is also absent in smooth muscle cell. And instead, there is a special protein called as calmodulin is present in smooth muscle cell. Also, smooth muscles structure includes dense bodies. The dense bodies connect thin filaments or actin filaments to each other instead of Z disc, which usually seen in skeletal muscles and cardiac muscles. For detailed description of differences between skeletal, cardiac, and smooth muscle, please listen to snippet number 6, in which I will cover these differences in depth. Now, with regards to smooth muscle organization, usually there are two different types of smooth muscle fibers. One is the single unit fiber and the second is a multi-unit fiber. Now, single unit fibers are usually more commonly seen and they're present in blood vessels, gallbladder, urinary bladder, gut wall, ureter, bile duct, and uterus, while the multi-unit fibers are less commonly seen and they are present in ciliary muscles and iris muscles of eye. With regards to fiber coupling, single unit fibers are joined by gap junctions anatomically and also they're electrically coupled with each other, while multi-unit fibers are not joined by gap junction. Single unit fibers are normally stimulated either via autonomic nervous system, that is sympathetic or parasympathetic stimulation, hormones, or local factors, while the multi-unit fibers require stimulation either via autonomic nervous system or hormones. The single unit fiber contraction is usually slow and steady, while the multi-unit fiber contraction is asynchronous. Now, coming back to the actual contraction process, the contraction involves cross bridge formation similar to skeletal muscle and cardiac muscle. However, this is mediated by calmodulin instead of troponin tropomyosin complex. So the actual process, so once the membrane is depolarized, it activates L type of calcium channel, which are voltage gated channels. Calcium channels open, that leads to extracellular calcium moving into cell along the concentration gradient. This further leads to intracellular calcium binding with calmodulin that forms the calcium calmodulin complex. This further activates myosin light chain kinase or MLCK. Myosin light chain kinase activates myosin head via phosphorylation through ATP hydrolysis. Now, activated myosin attached to actin binding site and pull thin filaments together. Now, since these actin filaments are attached to dense body as discussed above, when thin filaments slide past thick filaments, they pull dense bodies together. That's how it leads to the entire muscle contraction. Contraction process continues until ATP-dependent calcium pump actively transports calcium ions back into sarcoplasmic reticulum. Now, low concentration of calcium always present in sarcoplasm to maintain the tone of the muscle, and that's why the smooth muscles are normally under constant tone rather than in a relaxed state. With regards to relaxation process, it requires myosin head to become dephosphorylated, and this process is mediated by myosin light chain phosphatase. That's how you need to describe the contraction and relaxation of the smooth muscle. I will be back in week's time with another primary snippet, and I will cover the differences in smooth muscle, cardiac muscle, and skeletal muscle in detail in primary snippet 6. Till then, goodbye and have a nice time.